Today we're talking about DNA cloning, how restriction enzymes work, how ligase and plasmids work, and how DNA clones itself in bacteria. DNA cloning starts with the specific gene that we want to clone. So for this specific gene, what we have to do is we have to first isolate it with restriction enzymes. Once we isolate the desired gene using restriction enzymes, we will be left with overhangs on the desired gene known as sticky ends. We can use these sticky ends to paste the desired gene into a plasmid and solidify that bond using DNA ligase. Now diving a bit deeper into restriction enzymes have two main purposes, which can technically be counted as one, but I'm going to separate them for the sake of the video, and those are protecting a bacteria cell from foreign bodies and cutting out a desired gene. Once the virus injects its viral DNA into the bacteria cell, the bacterium must quickly defend itself to survive. One of the key defense mechanisms it uses is restriction enzymes. Bacterial DNA often is chemically marked with methyl groups at specific recognition sites. These methyl groups act as protective signals when a restriction enzyme scans the DNA. It can tell the difference between the bacterium's own methylated DNA and the invading unmethylated viral DNA. The enzyme will cut only the foreign DNA, leaving the bacterial genome unharmed. This system is called the restriction modification system, which allows the bacteria to selectively destroy viral DNA while protecting their own. By combining precise DNA recognition with protective methylation, bacteria can survive in environments full of viral threats. In essence, restriction enzymes are the defenders, and methylation is the safeguard that ensures only the invader is targeted. Now that we understand how restriction enzymes function in protecting bacterial cells, let's focus on its cutting aspect and look more closely on how this cutting ability is used in genetic engineering. So let's imagine we're working with a long strand DNA molecule, and somewhere within it lies a specific gene we want to remove. See, now we don't randomly guess where that gene begins and ends. Instead, we select a restriction enzyme that recognizes short palindromic sequences located on both sides of the target gene. Once the enzyme reaches its recognition site, it cleaves the diphosphoester backbone, cutting through both DNA strands with precision. This cut usually produces what we call sticky ends, short single-stranded overhangs that remain exposed after the strands separate. These sticky ends are incredibly useful. The unpaired bases are free to hydrogen bond with complementary sequences, which means they act almost as molecular Velcro. They make it much easier to connect the gene we've cut out to a new piece of DNA. Once the gene is excised, we typically transfer it into a plasmid, working as our vector. But before we insert the gene, we also cut the plasmid with the same restriction enzyme. This is a critical detail because cutting with the same enzyme ensures that the plasmid sticky ends are complementary to those on the gene we just isolated. As soon as the two come together, they align and base pair naturally, almost as if they were meant to join. By using restriction enzymes this way, we gain the ability to isolate a single gene from an entire genome and prepare it for recombination. This cutting process is not just a step in cloning, it's the foundation of recombinant DNA technology. After inserting our desired gene into a plasmid, we also include an additional gene. Typically one that provides antibiotic resistance. This step is crucial because when the plasmid is introduced into a host cell for cloning, many colonies will form. However, not all of them will carry the gene we're interested in. So, by adding antibiotics to the growth medium, only cells that contain the plasmid and therefore the resistance gene will survive. This is a selective process that allows us to easily identify and isolate the colonies that actually contain our desired gene, ensuring that the subsequent experiments will use the correct genetic material. DNA ligase is often called molecular glue, and this is because DNA ligase will seal any breaks in the sugar phosphate backbone that is made by our selected gene being put into our plasmid. This makes the recombinant DNA stable and keeps the inserted gene secure, making our plasmid able to carry our selected gene into whatever host cell we are taking it into.
In this part, I'm explaining how the calcium chloride method helps bacteria take up plasmid DNA. Bacterial cell membranes and plasmid DNA are both negatively charged, so they naturally repel each other. Because of that, DNA cannot enter the cell on its own. When we treat the bacteria with cold calcium chloride, the calcium ions neutralize the negative charges on both the cell membrane and the plasmid DNA. This reduces the repulsive action and allows the plasmid to move closer to the cell surface. The cold temperature also makes the membrane more rigid and stable. Once inside, the membrane reseals and the cell now carries the plasmid. Now the bacteria becomes transformed, meaning it has successfully taken in the foreign DNA. As it grows and divides, the plasmid is copied along with it, so every new bacterial cell will also contain the same plasmid. In this part, I'm going to walk you through the key steps in the cloning process using plasmids. We start with two things: foreign DNA, which contains the gene we want to insert, and a plasmid taken from a bacterial cell. Both the plasmid and the foreign DNA are cut using the same restriction enzyme. This creates matching sticky ends that can easily join together. The foreign DNA fragment is inserted into the opened plasmid. DNA ligase seals the sugar phosphate backbone, creating a single continuous loop. This gives us a DNA molecule, a plasmid that now carries the new gene. Once the plasmid is ready, it's introduced into a bacterial host cell using the transformation process. Now all bacteria will take up the plasmid so to identify the successful ones. The plasmid contains a resistant gene, so only the bacteria that actually receive the plasmid can survive and grow on this plate. The ones that didn't pick up the plasmid won't be able to grow. The surviving bacteria multiply again and again. Each cell contains the same plasmid, so as they divide, they create a many identical copies of the inserted genes. This results in multiple clones that all carry the foreign DNA, and that's the full cloning process, from cutting DNA to forming a plasmid, transforming bacteria, and finally producing many identical clones.